Hello everyone. In this video I am going to talk about microarray, specifically DNA microarray. We have different types of microarray and the type of microarray depends on the material which we use. For example, if we go with the DNA, we call it as DNA microarray. If we use RNA as a material, we call it as RNA microarray. Likewise, if we go with the protein, we call it as protein microarray. And we have tissue microarray as well. Please excuse me for the background noise, it's heavily raining here. Sorry for that, and let's get into the topic. Before we talk about the DNA microarray, first let us discuss what this microarray is and how it is produced and what is the use of microarray. This microarray is nothing but a microscopic slide that is either made up of glass or silicon. It contains an ordered series of a sample, and that sample could be DNA or RNA or a protein. Okay, and by using this microarray technique, we can detect the expression of the gene. We can easily make out which gene is expressed, like which gene is active and which gene is repressed. So this microarray allow us for the simultaneous interrogation of several hundreds and thousands of a gene. For example, if you take this as a microscopic slide. So in this microarray technology, at the center portion of this slide, there will be a small grooves where we are going to deposit the gene into those grooves. For example, if we take DNA microarray, we are going to deposit a small DNA spots into these microgrooves, where this microscopic slide can accommodate around 6,000 to 6,500 genes, especially in this center region of a microscopic slide. Now let's see how these microarrays are produced. It is not at all easy to prepare a microarray. It's a combination of art and science, and moreover, to prepare a microarray, you need experienced people from different fields, like you need a chemist, you need an engineer, you need a molecular biologist. And the aim during printing is to produce reproducible spots with a consistent morphology. As I told you just now, we are going to deposit the genes into those grooves of a microscopic slide. So here we are going to place the DNA in the form of a spot. So we are going to produce a reproducible spots with a consistent morphology. It's same like a printing technology. Like if you give a print of a document, we are going to fill the cartridge with the ink. So this ink will be deposited onto the paper in the form of a letters, right? So once you select the document and when you go for a printing, you are going to get a printed document with the letters printed on it, right? So it resembles the same. Here we are going to deposit the genes, nothing but we call it as a DNA spots onto the microscopic slide, and that will be utilized as a probe. I think you are familiar with the Southern blotting technique. What we do in the Southern blotting technique? So once we separate the DNA using a agarose gel electrophoresis technique, then we transfer those bands onto a nitrocellulose membrane, and then we apply some radio labeled probes. And if these probes have a complementary sequence with the DNA, they both will hybridize with each other, and we can easily detect that hybridized region, right? So here this microarray technique is also based on the same principle. And moreover, with this technology, we can study the expression of some hundreds and thousands of genes at a time. I told you this is the microscopic slide, and at the center region we are going to deposit the DNA as a spots, right? So this entire microarray will represent the complete genome of a particular organism. You can choose any model organism of your choice. You can choose yeast as a model organism, you can choose human as a model organism, you can choose mouse, you can choose rat, whatever. That's what I said. You can customize these microarrays based on your experimental needs. This microscopic slide will act as a probe. Here we know the sequence information. Now what we are going to do in the next step, we are going to add test sample to this slide. And this test sample will contain a DNA with unknown sequence. Now when we apply this unknown sequence onto this slide, this DNA which is present in the experimental solution will go and bind with this sequence if they both are complementary to each other. For example, if we look into a single groove, here we have added a gene that carries a sequence of ATGC. Now we have added a test sample that carries a sequence of TACG. Now both are complementary to each other, right? Because adenine pairs with thymine and guanine pairs with cytosine. Now this sequence will go and base pairs with this sequence ACG by forming a hydrogen bonds. Earlier it was a single stranded DNA. After the hybridization it converted to a double stranded DNA. Now we are going to detect whether the hybridization has taken place or not. That can be detected with the help of fluorescent markers. Once these microarrays are synthesized and it is given to a buyer, these microarrays should be stored in a dry condition. That is, they should be away from moisture, and they should be stored in dark conditions, that is away from sunlight. And moreover, you need to store these microarray slides at a temperature of 20 degrees centigrade, or you can leave them at room temperature. So you will be given with the instructions of storage. So based on that you need to store those microscopic slides. And the shelf life of this microarray is only 6 months. Coming to the principle, as I told you just now, this microarray technique is based on the principle of hybridization. Hybridization between the probe and an experimental sample. See here, this is the microscopic slide. Now at the center portion we have a DNA spots, okay, each with a specific sequence, right? So this microarray will represents the complete genome of a test organism. For example, we take yeast as an example. Now this microarray will carry all the genes of a yeast organism, right? See here, this slide will now act as a probe. We know the sequence information that is present in the each groove of a microscopic slide. Now we are going to apply the test sample. For example, I have two samples, yeast sample 1 and yeast sample 2. Okay. Now I want to find out which genes are active and which genes are repressed. So, one of the yeast organism is growing at aerobic conditions and one of the yeast organism is growing at anaerobic conditions, right? Aerobic means it requires oxygen for its growth, whereas the anaerobic means it doesn't require oxygen for its survival. So one of the yeast organism is growing under aerobic conditions and the other organism is growing under anaerobic conditions. Now I want to check which genes are responsible for its survival in both the aerobic and the anaerobic conditions. What I will do is I will use these yeast cells for the isolation of RNA. I am going to isolate RNA from both these yeast cells, right? Then I am going to convert this RNA into cDNA, that is complementary DNA, with the help of an enzyme called reverse transcriptase. Now I have two DNA samples, one that is isolated from the yeast which grows under aerobic conditions and the cDNA from the another yeast sample which grows under the anaerobic conditions, right? Now I am going to add a fluorescent marker to both of these samples. So I use green fluorescence here and I use red fluorescence here. So now the cDNA is fluorescently labeled. See the difference here. The yeast cell which is growing under the aerobic conditions is labeled with a green marker. So this is how it looks like. Now the yeast cell which is growing under anaerobic conditions, after the extraction of RNA followed by the synthesis of cDNA, I am going to add a fluorescent marker which is red in color. Now this cDNA is tagged with a red fluorescent marker. Now you can easily make out the difference. So this is green in color and this is red in color. So in the next reaction, what I do is I am going to mix both these samples, right? This is how it looks like. So this is the cDNA that is from the anaerobic yeast cell, and this is the cDNA from the aerobic yeast cell. Now my sample is ready. This is the experimental sample. So I told you earlier, there should be an hybridization between the probe and the experimental sample. So this is now a probe with the complete genome sequence of an yeast organism. And now this is the experimental solution which contains a combination of two cDNA. That is one from the aerobic yeast cell and the other from the anaerobic yeast cell. Now I want to check which genes are active and which genes are repressed. Now I am going to apply this sample onto this microscopic slide. And next step is I need to look for the hybridization. After this step of hybridization, for example, look into a single spot. If it carries ATGC sequence and if our experimental sample carries a gene with a sequence of TACG, both will base pair with each other by the formation of hydrogen bonds and they will hybridize with each other. That means they will stick together. Now I need to find out whether the hybridization has taken place or not. Following this step of hybridization, we need to go with the collection of data and the analysis of data. Now we are going to look into the each spot. As I already told you, we are going to place some hundreds and thousands of genes here. For example, this yeast is having around 6,000 genes. All these 6,000 genes will be incorporated onto this microscopic slide. And now we have applied the experimental sample, right? Now when we analyze the data, if you look into the data, some spots will appear green, some spots will appear red, and some spots will express both green and red. That gives you some yellow or orange color, right? And there will be some spots without any color. That means there is no expression of gene over there. And if you look at this green spot, that indicates that the gene under the aerobic conditions is expressed more. Whereas if you look into this spot, here there is a red color. Red color is for the anaerobic condition, right? That means the gene from this yeast that is growing under the anaerobic conditions has expressed more compared to the yeast cell that has been grown under the aerobic conditions. If you observe the expression of both the genes, then it gives a different color. So based on the color intensity, we are going to check for the expression of the gene. Here in this probe, we know the sequence, but we don't know the sequence here. But there should be a hybridization between the known and the unknown sequence. So based on that hybridization only, we can go for the analysis to check out which gene is active and which gene is repressed. Coming to the types of microarray, there are two types, that is cDNA microarray and a oligonucleotide microarray. Here we use cDNA, that is usually 600 to 2400 nucleotides in length. And if we want to make a multiple copy of that cDNA, we go with the PCR, where we are going to amplify the gene. And coming to the oligonucleotide microarray, here we are going to synthesize the oligonucleotides directly onto a glass slide. And that usually ranges from 20 to 50 nucleotides in length. And these are the short known nucleotide sequences, so there is a less chance of error. In cDNA microarray, what we actually do is, first we need to choose a source, like whether we go with a cell or a tissue. Then we need to go for the extraction of RNA. After we extract RNA, we are going to synthesize cDNA by an enzyme called reverse transcriptase. And once we are done with the synthesis of cDNA, we use a fluorescent markers. We can either label them directly or indirectly. After the labeling process, we go with the hybridization. After the hybridization, we go with the collection of data and the analysis of the data. And now let's see how this oligonucleotide microarray is synthesized. We synthesize oligonucleotide microarrays with the help of photolithography. See here, observe the picture. See, for example, this is the glass slide, center portion of a microscopic glass slide. Actually, I took few squares. It will have more number of squares, but for better understanding, I took only few squares, right? Now, this should be covered with a mask. So, masking is the key step of this microarray technology. So, when this slide is covered with a mask and we expose it with the light source, and this mask will make a specific pattern of light onto this solid surface where the light activates the surface. For example, this is the glass slide, okay, and this is the center region with a small grooves where we are going to deposit the DNA as a spots, right? Now we are looking how we are going to synthesize a oligonucleotide microarray, right? So initially what we do is we use a chemical as a blocking agent, and we are going to add this blocking agent to all these grooves, right? So the name itself indicates that it is going to block the addition of a new nucleotide, right? We have 4 nucleotides, right? Adenine, guanine, cytosine and thymine. So we are going to add one nucleotide after the other to make a chain of 20 to 25 nucleotides. Now these all grooves were added with a blocking agent and we cover it up with a mask. And when you apply a laser rays or when you give a light source, that is going to remove the blocking agent. So this specific area will get activated with the illumination of light. See, if you observe here, see, this is the pattern, right? Right? This mask should be exactly same with the center portion of this slide. And when we expose it with the light, that makes a specific pattern onto this solid surface, where this light is going to inactivate the blocking agent and it is going to activate these surfaces. Now you can add a nucleotide. For example, first we go with the adenine, and this adenine will carry a blocking agent again. Now this adenine will be incorporated here carrying a blocking agent. So we are stopping the reaction there with the addition of a blocking agent. Now we are going to change the mask pattern. Again we are going to add a second nucleotide, that is guanine. See, initially when we added adenine, all these exposed regions will be added with the adenine carrying a blocking agent. Right? Now we can't add another nucleotide here because this adenine is carrying a blocking agent again. Now we change the pattern of the mask. Again when we expose it to the light source, now it is going to expose these areas. For example, now we add a guanine that again carries a blocking agent. In this way we are going to add all the 4 nucleotides till each groove should have around 20 to 25 nucleotides in length. This is how we actually prepare a microarray. I will explain you in detail with the diagrammatic representation. Look into the picture carefully. This is the glass slide. So here these are the coupling agents and these dark colored ones are the blocking agents, right? So once you apply a light treatment, what happens? These blocking agents are photosensitive. That means they get inactivated when you expose them to light. So once you apply the light treatment, see, observe here, this is the mask. So the mask will make a specific pattern onto a solid support. These are the regions that are now exposed. Look here. So this is the mask, this is the mask. So this region is exposed, this region is exposed and this region is exposed. And carefully observe here, there is no blocking agent. The blocking agent is removed after you treated with a light. This light source is going to inactivate the activity of a blocking agent, whereas it is going to activate the exposed regions. Now in this So this is the first pick. In the second pick, you can observe that these coupling agents are now revealed. Now we can add a new nucleotide to that region. See, the rest of the regions were covered with the mask. The mask will stop the penetration of light at these regions. So as I told you earlier that this mask will make a specific pattern onto the solid surface, right? So here only these three regions were exposed, and those regions, after exposure to the light, the blocking agent got inactivated. Now what we do? We are going to add a nucleotide. So out of A, T, G, C, first we have chosen to add a guanine. Now this guanine is carrying a coupling agent along with the blocking agent, right? When you add that nucleotide, where it will go and bind? It will go and bind to these exposed regions only, not to the other regions. Now look at the third picture. Here the guanine got incorporated. Here the guanine got incorporated, and here the guanine got incorporated. And this guanine nucleotide is again carrying a blocking agent. If you want to add another nucleotide, see for example now the guanine got attached. If you want another nucleotide of thymine, or if you want to add another nucleotide of adenine to form a chain, again this blocking agent should be inactivated by exposing to a light rays. Once if you expose it with light, this blocking agent will be removed, and then the other nucleotide will be added over here. In this way, we are going to change the pattern with the help of a mask, and we are going to add one nucleotide after the other. In this way, we are going to synthesize the oligonucleotide microarray. Hope you are clear. See, look at this picture. These are the grooves. For example, first time this 1, 2 and these three regions were exposed, and we have added guanine here, here and here. Now we have changed the mask. Now the pattern is changed. Now the exposed regions is 1, 2 and 3. What we do? We expose it to light, and then we are going to remove the blocking agent here, here and here. Now what we do? We are going to add another nucleotide, say for example thymine, that again carries a blocking agent. Now when we add thymine, so these three regions are exposed now, right? Remember again these regions were closed again after the incorporation of guanine, and moreover they carry a blocking agent, so there is no chance of adding the nucleotide directly. Now we change the pattern of a mask. Now these regions are exposed. Now we are going to add another nucleotide, that is thymine. Now in these regions thymine will go and bind. In this way we are going to fill this grid with all the 4 nucleotides, and we are going to elongate the chain. So now each cell will have a specific sequence that is around 20 to 25 nucleotides in length. Once we are done with the synthesis of microarray, what we need to do? We need to go with the hybridization step, and before we go with the hybridization, we need to label the sample. So after labeling, we go with the hybridization, and after hybridization, we go with the analysis of the results. Coming to the steps involved or the actual procedure of this microarray technology, the first step is the collection of sample. Either you can use cells or you can use a tissue, and you can choose 2 different samples, that is one should be a healthy cell and the other one should be an abnormal cell, or here we can use a cancer cell. So the next step is the RNA isolation. You can isolate RNA by going with the kit method, or you can go with the phenol chloroform extraction method. As a part of RNA isolation, you need to specifically isolate mRNA only, not tRNA or rRNA. So for that reason, you can go with the use of column beads that carry poly-T-tail. So what happens when you use these columns that contain a poly-T-tail? We all know that mRNA will have a poly-A-tail. That means it will have a adenine residues at the 3' end, right? So this adenine, which is complementary to the thymine, will go and base pairs with the thymine. So it will be sticked with each other. Now in the next step, you are going to rinse those columns to collect specifically mRNA. In this way, you are going to isolate the RNA. After this step of RNA isolation, you are going to create cDNA labels. That means once you are ready with the RNA isolation, what you need to do? You need to use an enzyme called reverse transcriptase, and then you are going to get cDNA from RNA. As I told you, the synthesis of cDNA from RNA is called as reverse transcription, and that is carried out with the help of an enzyme called reverse transcriptase. Now you have cDNA, right? So the cDNA that is synthesized from a healthy cell and the cDNA that is synthesized from an abnormal cell. Now, as a part of labeling this cDNA, you can use different fluorescent markers like CY3, CY5. Here the CY stands for cyanine 3, cyanine 5. Or you can go with the CY 3.5, Tamro and also Texas red. Most commonly we use CY3 and CY5. So in order to tag the cDNA from healthy cell, we go with the CY3. And in order to tag this abnormal cell with a fluorescent marker, we go with the cyanine 5, which usually gives red color. This will give green color. So we can easily make out the color variations, right? Green and red. What we do in the next step is we go with the hybridization. After you are done with the preparation of sample, you are going to apply this sample onto a microscopic slide that usually carries a DNA microarray. Now after the hybridization step, you are going to collect and analyze the data. Now I'll explain you in detail with the diagrammatic representation. Just look it here. We are going to collect the sample as a first step, right? We can choose cells or a tissue. This is the healthy cell and this is the abnormal cell. Now what we do, we are going to culture this cell. So first here we have grown the healthy cell separately and we have grown the infected cell separately. In the next step what we do, we are going to isolate RNA. As I told you just now how we isolate the RNA, right? See, this is the RNA that was isolated from a healthy cell and this is the RNA that was isolated from an abnormal cell. In the next step, we are going to convert this RNA into cDNA. For that reason, we use an enzyme called reverse transcriptase. And moreover, we go with the addition of a fluorescent markers that we simply call it as tagging. See, observe here. Here we use CY3, which is green in color. And for the abnormal cell, we use CY5, which is red in color. So this is the cDNA that is tagged with a CY3 fluorescent marker. And the cDNA that is synthesized from the abnormal cell is tagged with a cyanine 5 fluorescent marker, which is red in color. Now you can easily make out the difference, right? Now in a solution, what we do? We are going to mix both these samples. This is the cDNA of a healthy cell and this is the cDNA of a abnormal cell. We are going to mix both these samples, making a single solution. Now we apply this experimental solution, which is in a liquid form, onto a solid support of a microscopic slide at the center region. So this is the microarray slide, which is carrying a set of genes. So this represents a probe. So here this microscopic slide represents the complete genome of a test organism, right, which will carry some hundreds and thousands of genes over here, right. After we apply this solution onto this microscopic slide, we look for the hybridization. When will the hybridization take place? Only if these strands that carry a complementary sequence with the sequence that is present in these grooves, then they both will stick together, forming a hydrogen bond that we simply call it as hybridization, right. So after this step of hybridization, we go with the collection of data and the analysis, right. Now this microscopic slide is ready, right. You have applied the experimental sample over here and you allowed for the hybridization. After this step of hybridization, remember you need to wash this slide to remove any unbound regions, right. So now you are going to look for the hybridized regions, right. So once you are done, you are going to place this slide in a scanner. That scanner will contain laser, computer and a camera, Once you place this microscopic slide in a scanner, that scanner is going to scan this microscopic slide. Laser will excite the fluorescence of cDNA. See, this cDNA is carrying a green fluorescence and this cDNA is carrying a red fluorescence, right? So that laser which is present in a scanner will excite the fluorescence of a cDNA. While this laser scans this microscopic slide, that information will be recorded by the camera that is present in a scanner, and that camera will store the information and that will display on a computer, immediately giving you the result. Whatever the data you get, that you are going to analyze it now, right? So if you look into this picture as a part of analysis, now you will get a picture like this. Some of the spots will show green color, some of the spots will show red color, and some of these spots will show a combination color. That is due to the expression of both the cDNA from the healthy cell and from the infected cell. Some of the regions that fluoresce yellow or orange color. That is due to the combination of this green and red, right? When you interpret the data, like you can easily make out with this color. The green indicates that healthy sample is hybridized more compared to the abnormal sample. When coming to the red, see here, the red color is given to the cDNA that was isolated from the abnormal cell. When this sequence go and hybridize with the probe, that we can clearly make out here with the appearance of the red color. So red color indicates that the abnormal sample hybridized more than the healthy sample. If you see a combination of yellow or an orange color, that is due to the expression of both the samples, that is both the abnormal sample and the healthy sample. Both of them got hybridized to the probe. This is how we make out. Based on the intensity of the color, we can easily determine the activity or the character of a gene. Now let's discuss about the advantages and the disadvantages of microarray technique. Coming to the disadvantages, it is very expensive method, and moreover it requires expertise to perform the analysis. And one more disadvantage is that here we have involved some thousands of genes and we are going to check out the expression of those genes, right? There will be a large data incorporated and that takes long time for the analysis. And one more disadvantage is that these microarray slides can be stored only up to a period of 6 months. That is, the shelf life is very less. Coming to applications, this DNA microarray can be used for the study of gene expression and profiling. As I told you just now, how we check for the expression of a gene. And moreover, it is used in the diagnosis of a disease. And once we diagnose the disease, we can go with the treatment options also, like we can check the efficacy of the drug using this technique also. See here, the another application is gene discovery and as well as the drug discovery. We can check different drugs efficacy with the help of this technique, and moreover it is used in the field of research. This is all about the DNA microarray. Don't get confused. This DNA microarray can be also called as biochips, DNA arrays, gene arrays, DNA chips and gene chips. Whatever term you use, that is related to DNA microarray. In my next video, I will talk about protein microarrays. Take care. Thank you.